Welcome all of you to join today's presentation of the summer research program for medical students. The program is organized by the Foundation of Health Science and the National Health Research Institute, NHRI. This year, we have a total of 35 medical students applying to this program. And whatever you have accomplished with this, it definitely will go a long way to contribute to better knowledge of medicine. And this is the most important goal and spirit of this program. Thank you all.
Okay, welcome to the 70th conference of Summer Research Program. So I would like to introduce the first speaker, Sakai He, 6th grade medical student from National Taiwan University. The advisor is Professor Xie Yiting from Department of Ophthalmology.
We can see that on the right-hand side, it is a close-up view of our OCT image. So previous work shows that if there is some defect in fovea region of our retina, we included patients with long-term HCQ treatment from 2006 to 2022. We conduct the principal component analysis. And it turns out that RP has also have the highest goodness of fit.
Are you convinced that your finding is truly representing the progression pattern from peripheral to central? Because cross-sectional study is not able to establish the causal relationship.
Because this is more like a observational finding, and we don't run any statistical estimation to look into that.
However, in the recent decade, the AHR is also shown very vital to immunoregulation in inflammatory disease and also autoimmune diseases. So as we find the target molecules, we can then correlate them to the fibrosis seen in the SSE-ILD, since it is the unmet medical need. For our results, as we mentioned in the microarray database, we did not see a significant change. Yeah, so we can take the expression of these molecules and draw a heat map. In the heat map, we can see the genes over here and the data sets or cells over here.
Can you go back to your IHC scanning? From this slide, in the sclerosing interstitial lung disease, did your data compatible with your IPA data?
It's because when we are constructing this animal model, we use the SSE-ILD model, and we actually analyze the fibrosis by using some Mason trichrome staining.
And CSD is the neuronal glial depolarization wave propagating in the brain tissue. During CSD propagation, it will promote the release of substances such as nitric oxide, arachidonic acid, potassium ion, and hydrogen ions. However, the potassium chloride-induced CSD undergoes microsurgery, so it remains the characteristics of invasion. We expect to see the lesser CSD frequency in our non-invasive VNS group.
What will be your clinical implication of your study? I mean, how do you translate the mouse study into human clinical use? In your study, the result of your study, how will you translate to human clinical use?
Our mouse model is for its efficacy of the transgenic mice, because mouse model provides more options for transgenic mice.
Heart failure is one of the most important cardiovascular events, which is associated with high mortality and hospitalization rate. Our study aims to identify some plasma protein biomarkers, and we eventually choose X3 boost as our best models because it has the highest mean AUC score. It first makes a decision process to predict a heart function status. And then we applied the SHAP explainer to see how predictions were made inside this.
From your data, the XG boost, the AUC value is only 0.795. Do you think if this value is adequate to persuade the clinician to use your machine learning data to predict the heart failure patient?
The hyperparameter tuning process is that it can change parameters at the process of building these models to make predictions more close to minimize the difference of the predicted labels and true labels.
The pathogenesis of T1D is influenced by several well-documented genetics and environmental factors. N-glycosylation is the enzymatic process to form linkage between glycans and proteins. We reanalyzed gene expression of N-glycan processing enzyme in PBMC of T1D patients from GEO database and analyzed pro-inflammatory cytokines proliferation and N-glycan branching in T cells of spleen.
Do you have any explanation of the viral accumulation increase and the bacteroids going down?
Yeah.
Is any related with the ketogenic diet? Any explanation?
Indeed, many researchers show that like toll-like receptor affected by microbiota, like toll-like receptor 2 and toll-like receptor 4, and MYD88 mutant will significantly inhibit the diabedogenesis in not mice.
I have the pleasure to introduce two parts of the student presentation. The first one I'm going to present is Chu Zizhen, and his topics today is pentraxin 3 as a novel therapy strategy for pulmonary arterial hypertension via the YAP and TEST pathway.
During the progression, pulmonary arterioles undergo vascular remodeling. We hypothesized that PTX3 antibody can suppress cell rigidity and inflammation. We first confirmed that TNF-alpha can trigger NRH3T3 cell line to secrete PTX3. Animal model, we can conclude that PTX3 can regulate the activity of YAP-TADs. We will use tissue fact cytometer to quantify IF standing of YAP-TADs on our animal model.
Do you know the normal physiological function role of PTX3? Is it expressed even under the normal physiological condition or not?
The CEBPD gene is upregulate of PTX3, and as our collaborators' data, we find out that CEBPD can upregulate PTX3. But however, CEBPD also have other effects on other pathways.
And to limit the ischemic size and reduce mortality. And the KCNQ is a kind of potassium voltage-cated channel. And ML213 is an emergent KCNQ4 opener. The rat will be anesthetized by intraperitoneal injection of uracil. And since the AST, ALT, and creatinine significantly declined, MIR-induced heart, liver, and kidney damage.
How to define the lesion? Is it induced by infection or by the MIR in the mice. The heart injury is induced by liver infarction or by the MIR. How to tell the difference?
We will tie the silk on the left anterior descending artery. And when we dye the events blue, we will tie the silk. So when the heart is still bumping, the events blue will go into the heart and through the other tight coronary artery. So we will know that the region that didn't have the events blue.
While a typical enhancer activates the expression of target genes. In this figure, you can see that there is a signal of MSH6 at transcription starting site, and echoing that MSH6 is important in the core promoter. After inducing the gene expression by IPTG. So to understand the interactions between H3K4 and the PWP.
What's the functions for the MSH6, both in the normal hematopoietic cell and also for the leukemia cell? Okay, if we deplete the MSH6, what will happen for the normal blood cell and also the leukemia cell?
When we knock down the MSH6 in leukemia cells, we found out that the cell growth will be decreased. The cells will die.
The first step is that the cells reprogram their metabolic strategies to sustain their growth and invasion. So this evidence is shown that MRC2 may be associated with gastric cancer metastasis. We found out that after knocking down MRC2, all the 4 enzymes were affected, especially SCD1. So it proves that MRC2 enhances SCD1 expression in gastric cancer cells. Further, in future work, we would like to know whether SCD1 is involved in the tumor formation in vivo models.
Tumor implants in genetic mouse, the tumor cell, whether they have the different growth resistance between the wild type and the MRC2 knockout mice. And secondly, that's since MRC2 have many effects, and they will have try to develop the MRC2 inhibitor in the near future.
Since MRC2 is an endocytic receptor, so it will move from the cell membrane to the endosome. And for many some method targeting the cell membrane proteins.
Typical circadian misalignment protocol or related research will have the outline shown on the screen. With this method, we will have a trained professional in our research team who recognize the pattern of different sleep stages or conscious stages. So 7 WKY and 7 SHR rats were randomly selected, and feature selected was EMG power, the amplitude of ectogram, and mean power frequency of electrocephalogram.
Just for curious, why do we need to discriminate WKY and SHR rats? And have you compared your algorithm to other many automatic scoring algorithms?
I myself think it's kind of less, so it doesn't show really promising results. So I took a step further to see if our algorithm can recognize the different kind of strains.
winner of our excellent award chose Temple University. The winner of our outstanding award is Taiwan National Defense Center. The winner of the award is 林世宏 National Medical University.